afternoon, everyone. Welcome to the Wageningen Graduate School Symposium that traditionally precedes the annual DS celebrations. Today we have as a topic the pandemics, and we have a line of young scientists lined up for you. I'm Karen Schrouwen. I'm the scientific director of the graduate school VLAG, and that is one of the 6 graduate schools operating within Wageningen University. We take good care of our young candidates by supplying them with a lot of information, high-quality courses, both content-wise and skills-wise, so that they're ready to make an impact, not just in the scientific field, but also in society as a whole. It's the first time that we actually do this symposium in an online setting. And the nice thing about that is also that we will reach more people than ever. Really, we have the largest number on board, and I welcome you all very cordially. As mentioned, we have 4 speakers for you that look at the COVID pandemic from many different angles that also represent Wageningen University. So what we will do today is go from pandemics as such, so transmission of diseases from animals to humans, to the very specific one that we have in mind at the moment, so COVID, and take it even one step further toward the social realm. So we'll also hear today about the effects on smell and taste, and also how people are affected in their daily lives in terms of food security. So it is really quite a wide spanning what we're going to hear this afternoon. I hope that all these topics are attractive for you. And in order for you to be also able to participate in this symposium, we have a live chat. So you can post your questions there, and I will get the questions, and some of them I can answer to our speakers today. And obviously it will not be possible to address all of them, but we will hand them through to our speakers so that they can answer them at a later time. All right. Now, without further ado, I think it is time that I start introducing the first speaker of today, who is Binneke Bron. She works on diseases that are transmittable from animals to humans. Obviously, a very important topic, and I'm really curious what she has to say about that.
Well, thank you, Karen, and nice to meet all of you. I wonder what you would have told me 2 years ago if I would have asked you, could a pathogen from an animal cripple the world? You would have probably said I was crazy, but here we are today, living it. And these pathogens that jump from animals to humans have been around for ages. And so behind me, you see a prairie dog. And I met this prairie dog about 10 years ago during my veterinary medicine research internship. And next to that, you see me again working on my PhD research, which was part of a large prairie dog plague vaccine project. And yes, I said it, plague, pandemic plague. The plague that killed nearly 1/3 of the European population in the Middle Ages. And this plague is still around. This pathogen is maintained in these wild rodent populations and still causes disease in them and sometimes in humans. But plague isn't going viral anymore because we know the pathogen that causes the disease. We know how to detect it and to diagnose it, how to treat it, and how this pathogen is transmitted, so we can prevent its spread when spillover from an animal to a human occurs. And so as we've lived with these pathogens for ages, we have developed many intervention tools, but somehow SARS coronavirus 2 outsmarted us. Or you could say, we were too slow to adapt, because we accidentally created this perfect network for pathogen shedding and spreading. As we and our domestic animals invaded a natural habitat, we put pressure on wildlife health, potentially increasing the chance of pathogen spillover to us and our domesticated animals. As we and our animals live in high densities, we're facilitating the spread from animal to animal and human to human. And as we, our animals and our animal food products, travel all across the globe, we spread a pathogen rather rapidly all across the world, as we're all experiencing right now. And so to prevent this from happening, we need to detect these pathogens really early and then have the right intervention tools at the right time, the right scale, and with enough force to stop the spread. And as you may gather by now, all of these things are connected. But let me reiterate, these infectious diseases don't just affect the person that gets sick or the animal that gets sick. These infectious diseases devastate ecosystems, disrupt our economies and livelihoods, as we're experiencing right now. So if you think about it, the health of our environment, our wild animals, our kept animals, and us, the two-legged animals dominating the planet, is all strongly connected. And this is where I do my research. I study zoonotic diseases at this interface between wildlife, domestic animals, and humans. And I ask questions like, how are these pathogens maintained? What causes an outbreak? And what intervention strategy might be best? At Wageningen University, I study Rift Valley fever virus, which is a virus primarily transmitted by mosquitoes, affecting livestock in Africa, which could spill over to humans. And the World Health Organization put this pathogen on its list of priority diseases for research because of its epidemic potential. It could spread from Africa to Europe, and we have limited intervention strategies to stop the spread and outbreaks. And so we don't even fully understand the pathogens that are already among us. And so that's like one of the reasons why we're studying them. But we're also studying them to be better prepared when similar ones emerge. So the knowledge that we gained on SARS coronavirus 1 really improved our ability to identify SARS coronavirus 2 and to develop the vaccine as quickly as had happened. And by now you might wonder, Bineke, if we know all these things and you can model them and we have all these intervention strategies, why is plague still around? Can't we just get rid of it? And the simple answer is no. When more animal species than humans are infected, we likely have a reservoir. And that means these pathogens, like plague, Rift Valley fever virus, and most likely coronavirus right now, they are going to stay around. And so we need to learn to live with it and mitigate the risk. Thank you.
Thank you very much, Bineke. That is quite a story that you have lined up for us there. So many pandemics are potentially out there, and we'd better prepare for it, right?
We should definitely take infectious diseases seriously. And unfortunately, it's taking this big pandemic for all of us to realize how important these infectious diseases are.
Yes, I couldn't agree with you more. But do you still have maybe some advice on what we can do to be prepared better for what's coming at us?
It's an interesting question, as we are in the middle of a pandemic, and I think we're still actively learning. But I think an important step we need to take is to reflect on what happened to us. So take a moment to see what went wrong. How did we get in this situation? So we should learn and reflect and take our lessons. In addition, sort of from my perspective, we should look upstream, right? We should see if we could maybe keep our wildlife habitat more pristine. We could invest or try to develop better detection strategies, and also potentially keep updating our biosecurity, right? So we have really good tools. We shouldn't forget, we have amazing tools. We're so much better prepared for a pandemic than we were in the Middle Ages. We know what is causing it. So those are the things. So I think we should keep investing and also be ready to respond as a global community. And so not as individual countries, but when we find this again, that we can have the interventions ready and be willing to do this as global citizens and respond as
No, exactly. That's a very important point because, well, the pandemic doesn't know boundaries, right?
No.
So we have to be prepared as like humans living on planet Earth to do something about it, and also really reach out to others to be able to do that. Now, I have another question for you, Bineke, as well, and that's coming through the chat. And it is like, how widely would your model system be applicable?
Oh, that's a great question. So I mostly study diseases that are transmitted through a vector. So a flea with plague, or a tick-borne disease, a mosquito-borne disease for Rift Valley fever virus. If you think about mosquito-borne diseases, they come with a special dynamic, right? The special dynamic, the mosquito transmits it. So you could take that very broadly and say, okay, our model system, we can use the models we develop and adapt them to other mosquito-borne diseases. On the other hand, you can also think about how mosquitoes could be. There's a lot of species of mosquitoes, so they all have slightly different feeding preferences, etc. So you could also say it's very narrow. But what I would like to say, Karen, is that with climate change, the ranges of these ticks and mosquito species are changing. So as we found West Nile virus for the first time in the Netherlands, we can expect more of these exotic diseases to start moving, and also for new tick species, for example, to come to the Netherlands and to
so basically what you're saying is that whatever we're seeing in different countries could be like something that is knocking on our door within not too long, and that we really need to prepare as humanity for that.
Yeah, we should definitely look inside and outside of our boundaries, yes.
Yes, exactly. No, but what I like about the fact is that we need to get together in order to really be instrumental in doing something about it. There is another question coming in, and that is, how do you think about the contact between humans and domestic animals? Which role is that playing?
So that's a very interesting question. So we have our pets, right?
And of course.
We sometimes forget that we get diseases from our pets as well. Our dogs can bring in ticks. You can get cat scratch disease from your cat. We keep animals.
But we love our cats, right?
Yeah.
So, yeah.
We keep animals for food. And in this process of food production, we've become really good about testing for pathogens that are very common, so that biosecurity allows us to be very efficient in our food production. On the other hand, our behavior, so our excessive consumption, also forces expansion of agricultural lands, which again could disrupt the natural habitats and also increase that contact again for that initial spillover from wildlife to domestic animals to occur.
So it's clear that
it's a very complex system, yes.
No, but we're in it together.
Yes.
And it's a huge challenge. So thank you very much for now for your wonderful insights. I will also introduce our next speaker now, who is Jurre Steens. Jurre is a PhD candidate. And I mean, we all know that doing a PhD project is, incredible enough. On top of that, he also has decided to start his own startup company, specifically on COVID-19. And I'm really curious about the story he has to share with us.
CRISPR-Cas, a technology that went from its discovery to a household name in a matter of years, even winning the Nobel Prize in 2020. So what does CRISPR-Cas research at the Wageningen University relate to the current COVID pandemic? Well, CRISPR-Cas is originally a bacterial immune system that helps fight off invading elements that are attacking these bacteria. Our understanding of this immune system has revolutionized biotechnology in recent years, mainly in its applications in genome editing, allowing us to change the very code of life. Of course, this tool is very excellent because it's able to very specifically look for a specific sequence of genetic information, which we can program it to do. Think of it like the search function in a text editor like Word. You're able to look for a very specific sequence of letters, DNA in this case, in many pages of unrelated text. My work at the John van der Oos laboratory focuses on a certain type of CRISPR-Cas system. Characteristic of this system is that it produces signaling molecules when it finds its target, setting off the alarm bells in the cell, as it were. Our fundamental understanding of this system has allowed us to take it out of its natural context and actually apply it in a test tube. We've developed this test and modified it and exploited it in such a way that it creates a fluorescent signal when it finds its target. Recent developments on this tool have led to a tool that's capable of detecting pathogens like harmful bacteria and viruses. So these developments, with these great results, have led to the startup company Scope Biosciences within the Wageningen University, which I started with 3 friends I've known since the beginning of my bachelor's. Together with the various research institutes within Wageningen University and Research, we've started developing diagnostic tools for various applications in the agri-food sector, but also in healthcare. At the start of the COVID pandemic, we were able to work with the Wageningen Bioveterinary Research Institute in Lelystad, one of the first institutes in the Netherlands that was able to set up COVID testing, conventional PCR testing, as the first in the Netherlands. With their help, we were able to do some pilot studies on relevant real-life COVID samples with promising results. Since then, we were able to work with TNO, University Medical Center Utrecht and Radboud to further test and develop our rapid COVID CRISPR-Cas-based diagnostic tool. Of course, a diagnostic tool needs to be reliable and fast, sensitive and specific, but it also has great additional value if you can take the sample in a fast and non-invasive manner. At the moment, we're able to provide point-of-care testing within 30 minutes with results reliable as PCR. Of course, with these results, we still face some challenges ahead of us, like the rules and regulations that come with making such a diagnostic test, as well as scaling up production to a level that's adequate for national coverage. Of course, we think that with large-scale rapid testing, we can help out in reopening our country, where testing at music festivals, sports matches and restaurants can allow us to do this safely. Therefore, we think that our vision, together with extensive knowledge and expertise within Wageningen University Research, we can also look beyond COVID, where if we look and monitor closely at upcoming diseases and detect where possible risks are and developing diagnostic tests preemptively, we can and will be prepared for our next pandemic.
All right, and you're really addressing that in many different ways. You're very fascinating how you are combining being a PhD candidate with your own company. Really, I find it exceptional how you do this combination. I've got some questions from our audience, and they're wondering how come your test is simply so much faster than whatever is around?
That's a good question. We combine 2 very powerful technologies. One technology that's able to amplify and make from a small amount of target sample, let's say COVID virus, a lot of sample. And we do the very specific detection with CRISPR-Cas. So we combine 2 characteristics of both of these technologies into one system that's able to do it very fast and accurate.
That sounds like music to my ears, really, because it has a very big potential in it to also impact all of our lives. I mean, now it's completely impacted because of the virus, but you could like reverse these effects by the fast test that you're developing. Really amazing. But if you want to do that, obviously, then upscaling and stuff like that is a very, very important thing. So is the technology that you're developing indeed scalable? How do you look at that?
Yeah, so it's very important to realize it's a powerful platform technology. So that means that we only have to change what we tell CRISPR-Cas to look for in order to to detect almost any pathogen we want. So 90% of the system is actually the same for any pathogen or virus we want to detect. So in that regard, scaling up is not that big of an issue. Of course, now with COVID, we need to scale up to a national level which for a small startup company is a big task. And
I
this
can imagine,
is something for which we bring the expertise from outside. We don't attempt to do this all ourselves.
I mean, the challenge is big enough as it is, right? Yep. I also have a question here coming through me through the chat, and it's from a different angle, because if you look at it from a society point of view, this constant testing requirement is also a little bit of a burden. How do you see that, and how to secure data and privacy and all these other things?
That's, of course, very relatable, of course. We're living it at the moment. I think what we're working towards, and what's also in the previous talk very clear, we need national and maybe worldwide attention to these things and have a system that we can react quickly and don't have this lag phase of the year we currently have. And I think we're all working together towards that goal to make it safe and fast for everyone.
Yeah, absolutely. And what I also like is the fact that you, by adjusting it slightly, you could be ready for the next one, right?
Yes.
So it's quite, well, omnipotent, if I dare say. So you can really look at a different category of viruses all through one and the same approach.
Yes, for sure. So this knowledge, that's the great thing about Wageningen University. We have expertise in the agri-food sector, but also the virology sector. So we want to combine all this knowledge and see the problem arising in these human and animal interfaces and not react when it's too late. So
what you're also saying is that you could potentially test much earlier to prevent an awful lot of the things that are happening currently.
That would be ideal, yes. Yeah.
Ideal, but yeah, I mean, as ideal as it will get, of course. Here, there's another question coming to me, and they say that, or the question is that CRISPR-Cas would be as reliable as PCR. There is already some questions about the reliability of PCR as such. So how do you see yourself in that respect?
Well, we try to position ourselves next to the golden standard in the whole world, which is currently PCR. We do not think we can now state we're better than the golden standard. We're as good as PCR.
And I guess that is also really a matter of further development. I mean, we've been doing so much learning on the job as it is, that it's also logical that you do not really have all the answers to all the questions that people can come up with right now. But as good as the golden standard, I think that is as good as it will get at this very moment. So thank you very much, Jure, for sharing your story with us. Now, let me also introduce our next speaker, who will take it from a very different angle. We already talked about society. Many people that experience COVID have a loss of smell and taste. And Albrecht Posma, our next speaker, investigates this. It is very impactful in people's lives, and I'm really curious what she has to say to us about this.
Imagine yourself waking up in the morning, you're ready for your morning coffee. You walk to the kitchen and switch on the coffee machine. When you take the first sip of your coffee, you notice that it tastes different than you're used to. And then you realize that you didn't smell it either. This happened to a lot of people during the past year, as smell loss is one of the hallmarks of a COVID-19 infection. Approximately 2/3 of all patients experience smell loss during their infection. As smell researchers, we know how big the impact of losing your sense of smell can be on your daily life. It does not only affect how your food tastes, but it also plays a role in social interaction. Imagine how it is to smell your own child. And in how safe you feel. For example, you're not able to smell smoke or fire anymore. And there are patients who were not able to do their job anymore, as they were a chef or a professional wine taster. Therefore, we decided we needed to do something. We very quickly set up a new, unique and worldwide research consortium, including researchers from 63 countries across the world, and also included researchers from Wageningen University. This is a very unique collaboration, and usually it takes quite long to set up a big consortium like this, but now we all had the same goal. We first launched a survey that was translated in many languages, and that was already filled in by more than 50,000 patients. And the first results were quite surprising. Usually, people who lose their sense of smell only lose their sense of smell, but now we found that also taste and trigeminal perception were affected. The specific smell of a cheese is perceived by your nose, and usually you only are not able to smell this cheese anymore. But now also taste was affected, and that's perceived by your tongue. So that's, for example, the bitterness of your coffee. And trigeminal perception is when you eat something really spicy or drink a carbonated drink. This is what distinguishes smell loss in COVID-19 patients from other patients that have smell loss. Moreover, we found that the smell loss was very sudden and severe. From one day to another, patients were not able to smell anything anymore. And this was not related to having a stuffed nose, like we know in patients who lose their sense of smell due to a cold or a viral infection. This taught us that losing your sense of smell suddenly is a really important hallmark for detecting a COVID-19 infection. When you lose your sense of smell, you should stay at home and get yourself tested. Most patients recover their sense of smell within 4 to 8 weeks after they lose it. But there are also patients who suffer from smell and taste loss on a longer term. They lose their sense of smell for 6 months or maybe even a year. And there are patients who experience parosmia. This is a distorted sense of smell. When they smell their cup of coffee, it smells like vinegar to them. We decided we also wanted to investigate these long-term changes, and therefore we launched a smell and taste check. This check is a short test people can perform at their own home every day. They do it by using regular products, for example, fruit juice or shampoo. People have this available at their home, so they can also do the test when they're quarantined or in isolation. They smell and taste these products every day, and they rate how intense the taste or the smell is to them, so how strong it is, and how familiar it is to them. So in this way, we can measure how well they can smell it, but also if it's not distorted. This has two goals. Individual patients can follow up their own sense of smell over time and see how it might improve. And we as researchers can follow up long-term smell changes in patients with COVID-19 infection. In this way, we hope to get more insights in how we can treat these patients so that eventually we can let everybody enjoy their cup of coffee again.
That is, of course, also what I hope, very much so, Albrecht. I mean, many people do have a return of smell and taste, as you say. But for some people, I also know that it really takes an awful long time.
Yeah, that's correct.
And it's very impactful. So does it always return?
So the patients we now know who have the longest smell loss are the patients who lost their sense of smell a year ago, so that's when COVID-19 started. Mm-hmm. So these patients are still there with their smell loss, and what we know from the regular patients with smell loss is that there will always be a group. It's a small group, but there will be some patients who do not regain their sense of smell.
Ooh, that is really terrible.
Yeah.
Is there anything known about the mechanism behind the smell and taste loss?
Yeah, what we know so far is that the COVID-19 virus is in the nose, and it's not on the smell receptors itself, but it's on the cells that are surrounded or that surround these receptors, and they bind to the ACE2 receptors. So these receptors are at several points in the body, but also in the nose, and by affecting those cells, they hinder your ability to smell.
Wow. So it's actually not even that simple to try and mitigate what is going on.
Yeah.
What I also liked about your talk was that it can be really participative science. What we're talking about, we can basically do something ourselves at home and test our own sense of smell, and also send you the results, of course, because that would also help your investigations further. That's really a very powerful way of doing science. How did you come up with the idea to do it that way?
The research consortium was set up quite fast. I think over a weekend, emails were sent back and forth, and more people were activated and motivated to investigate this. And we thought of, the easiest or fastest way to reach as many people as possible. So then we first started with a questionnaire, but then we thought, okay, we need to involve patients also in the follow-up and also activate them and give them something back. So not only give us information, but also provide them with some information, also keep them motivated to repeat that smell and taste check. So you can do it every day, and then you can also earn some medals or points, so to keep
virtual credit points, I guess. No, but I mean, everything is very much into keeping people motivated, and bringing it home so much close to them, of course, is a way of doing that. Now, I got another question through the chat. There's also people that do not have smell and taste loss. Do you know what determines the difference between people?
Yeah, that's something we are investigating right now. So what we've seen so far is that most people who have really bad case of COVID, so patients at the intensive care, for example, they mostly don't have smell loss. So it seems that in some way it's also related to how you're infected or how badly you are infected. So we are not sure why or what or how exactly, but it seems that there is a difference between people. Yeah.
But I mean, it makes it even more prominent to do these checks and keep also a check on your own health, because this is an easy one. I mean, everyone can do it, right?
Yeah, it's really easy. Most people just do it, for example, in the morning when they have breakfast and think, oh, I'm drinking this fruit juice. Oh, let's do the smell and taste check. You can do it on your phone. And then if you make it a habit, it's really easy.
Yeah, exactly. And then that is also really what I like about the fact that you have this interaction going on. So it's not just this is the patient, this is the researcher, but you really get them into the entire story.
Yeah, and in our consortium, we also work together with patient associations. We also try to communicate our findings to patients and involve them in our research, so to also improve the way they receive information about smell loss, and so that they know what they can do at home to improve their sense of smell.
Wow. I think it's a really interesting story, Elbrich. Unfortunately, you also know that time is always running very fast in occasions like this. So thank you very much for sharing your story with us. And let me just go to the next and also the last speaker of today, who is Theresa Andreessen. She will take us to another part of society. Many people have lost jobs because of COVID, and because of that have gotten into financial issues, which makes them dependent on food banks and other social charities. I find it really amazing what she has discovered, because I already had a preview on the story, and I'm very happy that she's willing to also share it with us today. So Theresa, please, the floor is yours.
The corona pandemic is not only about the risk of getting infected. This pandemic and its associated measures also has substantial economic impacts. Businesses are closing, prices increase, and people experience income drops. As a consequence of these economic impacts, the need for food assistance has increased throughout the world. This has also been noticed by food banks in the Netherlands. They saw an increase in applications of clients during the first months of the pandemic. But asking for food assistance asks for courage and people tend to delay this step due to feelings of shame. Because of this hurdle, some food banks expect a follow-up increase of clients up to almost 50%. As asking for food assistance can feel humiliating and shameful, and a situation of poverty is already stressful in so many ways, a steady and resilient food aid system one can count on is very important. During the first months of the pandemic, the food provision by food banks has been challenged in several ways. They had to deal with varying food supplies due to stockpiling behavior in supermarkets, drop-off volunteers above the age of 65 because they have a higher risk for severe health impacts, and they had to adjust their food distribution practices to the measures, such as keeping 1.5 meter distance. Because of these challenges, together with the increased need for food assistance, my colleagues, Hilje van der Horst and Paulien Dekkinga, and myself asked the question, how resilient is this food aid system in the context of the pandemic? On the basis of interviews conducted by Paulien, we saw that the food bank system in the Netherlands was quite resilient to deal with these challenges. They adjusted the way they distribute food to continue their main function of providing emergency food aid. Some food banks changed from handing out food parcels to handing out gift cards for supermarkets. Others did spread food distribution over the week and gave clients a specific time to pick up the food. Besides these adjustments in their way of working, they also received a lot of support. New, younger volunteers applied. They received increased monetary donations, and they got support from national and local government. This increased support in times of such a crisis also emphasizes that food banks have become the dominant way in which we think about addressing food insecurity. Although our research has shown that the Dutch food bank system is quite resilient, there are still concerns. Together with food aid organizations and other stakeholders, we need to think about ways to ensure access to healthy food in a dignified way. Within my PhD research, I investigate in different ways of providing food aid in relation to the dignity of people who experience food insecurity. Hereby, you can think of offering product choice, fostering respectful social interaction, and organizing empowering activities. Within my research, I focus on improving short-term aid. At the same time, we have to keep reflecting on the bigger picture. As a society, we need to think about ways to prevent the need for food assistance. As Nelson Mandela once said, overcoming poverty is not a gesture of charity. It is an act of justice. It is the protection of a fundamental human right, the right to dignity and a decent life. This act of justice should be realized both in the ways we provide short-term food aid, as in policies protecting the human right to food.
Thank you very much, Theresa. My goodness, what a story again, and so impactful on people's lives. It's really amazing. And as you say, I mean, yeah, how to do this in a dignified way is, of course, a very important aspect. Yeah. Now, our food banks have a lot of experience by now dealing with the pandemic. Are there some specific good practices maybe that you can share with us?
Yeah, well, for example, an important thing that we have to realize because of this pandemic is that we have to prevent health inequalities to become bigger. And one part of this is making sure that everyone has access to healthy food. Mm-hmm. And food banks, for example, what they also did during the first months is that they accepted everyone who asked for assistance without, like, before they judged their eligibility.
So they were a little bit picky and choosy in terms of food to allow.
Yeah. Normally they have a intake process, and now they said because of the huge increase of applications, they immediately said, okay, you can get assistance, and we will judge later if you, are allowed to get that. But I think also at the same time we have to look around and see, and to take care of the people who don't get access to food banks, who are maybe also struggling to get food on the table.
No, exactly. It's much more maybe even than the pandemic. The pandemic just brought it to life.
Yeah.
That this is what is really going on.
Yeah, definitely.
I also have a question here in the chat, and I don't really have a name here, but what would be a dignified alternative for food banks?
Yeah, well, for example, food banks are, some food banks already changed from handing out food parcels to a supermarket setting. What I also saw, we also, Laura Vosse investigated in the experiences of clients during this corona pandemic. And clients really appreciated the supermarket vouchers they got because it respects their, product choice, and that every person has different needs. So, I think this is very important. And also, try to stay away from stigmatization within social interactions, and realize that, people who experience food insecurity are very different, have very different situations. So,
and there's many of them, many more than we maybe realize, right?
Yeah. It's still also a taboo to talk about it. So,
Yeah, so in that sense, it is such an important issue. I mean, I know I also said that to our first speaker, but it's not just us individuals. It's a country, and it's maybe even beyond countries. Important to really be aware of this, and also maybe take a bit better care of each other.
Yeah, I think it is an issue that should be addressed both on the community level, but also on a more structural level in policies. Making sure that everybody has access to healthy food. Yeah,
no, exactly. And the nice thing about the chat is that it's going berserk now. The question is like, what was the economic impact due to the virus and the disease it causes, or due to the social restrictions taken by the authorities?
And then the impact on?
The economic impact. So the economic impact, was that because of the disease, or was it because of all kinds of social restrictions?
Well, that's something I can't really answer, because that's not what my research is about. I really focused on food banks and food aid initiatives, and how they had to deal with these economic impacts. But I can imagine both.
Would also be my guess, but I'm just reading the question.
No, I can imagine.
Another question here is like, how can we learn from the situation that we're in at the moment to be more resilient toward the future?
Well, I think it is important for the future that we make a strong issue for policymakers to change structural things and to prevent people to become food insecure, and make sure in the future we not only depend on a charitable food aid system.
Exactly. So it is really something that needs to become very prominent in everybody's minds. And if that is the case, then we can also really do something about it.
Yeah.
Thank you so much for sharing your story with us, Theresa. All right, well, time flies when you're having fun. We've come already to the end of this symposium. Thank you so much for listening in. to wrap things up completely, I would really like to thank our speakers who have shown us that, well, COVID is one thing. It has many different facets, but it's also very much a global thing, and it concerns all of us. Within Wageningen University, we look at many different aspects of COVID now, and you see that we really contribute to making the situation better for everybody. So thank you very much one more time, speakers. And I hope you enjoyed the program, my audience. Thank you very much for your lively participation. And I can only wish you a very nice afternoon on my behalf and also on behalf of my speakers. But before I go, I would like to draw your attention to the fact that in 15 minutes from now, we will have the DIA Symposium. You can use the same link as you've done for the symposium, and then we will have another interesting program lined up for you, again on the same topic. It's still the pandemic. Thank you very much, and have a nice afternoon.
Highly esteemed colleagues, partners of all kinds, from government, from industry, from other academic institutions, from society, students, ladies and gentlemen, I would like to welcome you all very much to this Dies Natalis, the 103rd Dies Natalis of Wageningen University and Research. And this is a very special Dies Natalis, because it's for the first time since World War II that we have a Dies Natalis without the physical presence of a major audience. And it's also for the first time that we have a Dies Natalis with the Rector Magnificus without wearing his gown and his chain. We all know why that is the case. It was almost exactly 1 year ago, in 12th of March 2020, that the government suddenly announced a lockdown of all kinds of restaurants and bars, of all kinds of public places, but also of schools and universities. Of course, we had taken measures before, social distancing, hygienic measures, one-way walking routes, but a real lockdown came as a surprise to all. It was a wake-up call. Suddenly, as a high technological society, we were seeing ourselves in a situation where we were no longer in control. Of course, we had been in the situation before of a pandemic. Think about in history, for instance, the plague in the Middle Ages. In 1665, Isaac Newton had to leave his Trinity College in Cambridge, in the London area, due to the Great Plague in the London area. And he spent 1 year in social distance, we would say now, in the rural areas. By the way, that was a very productive year for him. But also later, we have seen major pandemics, SARS, for instance, or Ebola. We also know that we are still ill-prepared. This pandemic, COVID-19, came as a surprise. And we also know, by the same token, that we will see in the future an increased sequence of new pandemics or threats of new pandemics due to globalisation, for instance, or the way we live with our animals, major urbanisation, climate change, all kinds of factors which will increase the sequence of pandemics most likely. Over the past year, we have to try to cope with COVID-19. We've done that by assessing the major risks of social effects, health effects, economic effects, mental effects, and try to mitigate them also, especially, of course, lately by developing vaccines and a major vaccination program which we are now rolling out throughout society. Still, the past year really felt as living in a living lab, or living in what others would call real-world experiments. Because we were really experimenting in how to cope with this pandemic. And in coping with the pandemic, science played a major role. Let me make 3 observations on the role of science in the current pandemic. First, we saw that really the pandemic and the role of science in that changed the role, how we look at science. Science increased its legitimacy tremendously, and also the trust in science increased very much. We see that, for instance, in the role of science and scientists in the outbreak management team, and the way the outbreak management team advises, by scientific evidence, the government. Second main observation on science is that we see that the major acceptance of uncertainties has increased also. Acceptance by the wider public, but also by politicians. Think about, for instance, the curfew, how it was announced and how it could not really be predicted what the effects were of the curfew. So a major uncertainty, but was still accepted, both by politicians and also by society writ large. A third observation is also that science communication has increased, but also professionalized. In the major news broadcastings, we see increasingly scientists being present, rather than major media figures or politicians. Today, at the current Dies Natalis, we want to focus on the role of science in coping with a pandemic. Not only COVID-19, but especially also the threat of future pandemics. What role can it play in preventing pandemics, or in predicting them, or if not, preparing better for future pandemics? What do we know already scientifically, and what are the questions which are still out there? What kind of research do we need? It are these kind of questions which play a central role in the current Dies Natalis.
Good afternoon, and thank you, Arthur Mol, Rector Magnificus, for the opening of the 103rd Dies Natalis. My name is Marcia Luiten, and I'm your host for this afternoon. And together with our guests, we will discuss the major task for scientists in pandemic prevention, prediction, and preparedness. At my table, I'd like to welcome Henk Bekedam, former chief of World Health Organization in China and India, Marion Koopmans, professor in virology at Erasmus University, Emilie de Vet, professor and chair on consumption and healthy lifestyles, and Wim van der Poel, research leader on emerging and zoonotic viruses. Welcome to all of you. So we have the honor to discuss pandemic prevention, prediction, and preparedness with world-leading scientists. But first, I'd like to give the floor to a young and upcoming talent, environmental historian Daniel Gallardo Albaran. He obtained a doctoral degree in economics from the University of Groningen, where he studied the evolution of health, well-being, and inequality in a historical perspective. In 2019, Daniel joined the Rural and Environmental History Group at this university as a postdoctoral researcher. Currently, he's conducting a project on global health inequality, epidemics, and the diffusion of sanitation since 1850.
The COVID-19 pandemic has shown the far-reaching challenge that infectious diseases pose for a globalized world. Governments are struggling to control the epidemic by making hard decisions about the economy, healthcare, and individual freedoms. From a historical perspective, these challenges are not new. In fact, we have been here before. Take a look at the picture on the screen and see the concern in people's faces as they were forced out of their houses for disinfection purposes. This happened during a plague epidemic in Portugal more than a century ago. At that time, the expansion of railways and steamships made the world smaller. People could travel anywhere. But for this, we have to pay a price because viruses also traveled with us. This connection between globalization and disease became evident around 1900 during the third plague pandemic. This illness caused panic everywhere because it could kill about 50% of those infected. Also, there were no effective methods of treatment or prevention. In one of my projects, we are reconstructing the weekly diffusion of plague for dozens of cities to understand how diseases travel with people. As the map shows, Hong Kong experienced an outbreak in 1894 when infected rats passed the disease to humans via fleas. 2 years later, plague reached Bombay through trade, and from there it spread to the whole world. Just by 1900. As with COVID, trade hubs and migration routes played a key role spreading the pandemic. Quarantines, disease reporting, and quick action were crucial, but often government policies went beyond stopping the epidemic. Consider the case of Chinatown in San Francisco, which was quarantined using a barbaric method with barbed wire fences. Clearly, the official discourse of disease containment did mask inequality and discrimination towards the Chinese, who had little political voice. Eventually, the plague pandemic was conquered and then forgotten. The aim of my research is to recover our collective memory of epidemics, to enrich public health discussions with lessons from history. For instance, the case of plague shows that globalization has a flip side and that we should be prepared for future health crises. Also, access to health infrastructures should be equal for everyone to avoid any type of discriminatory policies that end up hurting the most vulnerable. This was essential to respond and build resilience during epidemics in the past, and it continues to be indispensable nowadays. Thank you.
Thank you, Daniel. Henk Bekerdam, you've been listening. Danielle said the challenges are not new, but we forgot the lessons we learned from previous pandemics. As a WHO chief, you were at the front line of the SARS virus in China, recent history. What was an important lesson you learned there in handling the virus outbreak?
Thank you, Masha, and thank you, Daniel. I think one of the most important part is about pandemics that you learn from it. First of all, it is something from, if you have a novel virus, it is sailing while building the ship, because that's very clear. You have a new virus and then you start discovering things. That's already something in itself. But it's also important that you not only learn from things, but that you also do things better. I feel very proud actually about, if I look at the Asian countries, those who have experienced SARS, they were better dealing with the SARS, with the COVID right now, and they are still better dealing. And how did they do it? They started also acting. They started strengthening their surveillance system. Their surveillance system, not the monitoring system, and the early warning system. They started developing an information system in which information could be shared, an integrated information system. And they started on something else that is very important. They started building capacities. That if there would be a pandemic, that they would have the capacities to follow up with things like test, trace, isolate patients, and putting patients in quarantine.
So, and that's obviously something we were lacking, this preparedness. Huh? Is there anybody at the table who has a question for Danielle?
Yes, thank you for a very interesting pitch. So there, throughout the history that you show, the time of travel has really decreased, which really has an impact on, can you travel within an incubation period? Can we somehow bring that back? So can we use that kind of information in our redesign of what we should be looking for in the future?
Yes, I think so. That is a critical element, and it was a critical element back then. Actually, the improvements in maritime trade really connected different disease environments in different countries. And this is, in a way, a blessing because it allows us to interact with people in the whole world. But it can be a challenge when a health crisis take place in a country, and then within the incubation period, that person which comes from the, let's say, from the area that has suffered the crisis reaches an untouched population. This is definitely something that basically explains why plague became pandemic in 1900. And I think this should tell us that these, that the benefits of globalization, while for most people are very good, it also comes, with a different, darker, perhaps more problematic side that we should take into account in order to, build up resilience for future health crises. Because definitely, traveling times are getting shorter and shorter.
Okay, thank you, Danielle Albargaran. While we're still dealing now with COVID-19, new viruses are upcoming, and the question is not if, but when we will face the outbreak of a new virus. With our guests, we will discuss how we can handle the next virus in a better way. And our question is, what is the role of science in the prevention of a pandemic? Because we live in a world where there are more people than ever, more livestock than ever. Many of us like to travel, as you just mentioned. Wim, you're an expert on zoonosis. Have we created an environment that is favorable to zoonosis?
Yes, I think we have more or less created an environment where there is an increasing risk of zoonosis. When you look at the world map and when you look at the number of emerging zoonotic viruses over the world, there are certainly hotspots of zoonosis. And these hotspots are often areas where a lot of people live together in high density, also close to animals, often livestock, but also wildlife, where people are more encroaching into their environment. And that means that there are more regular contacts of people with specific species, and that means that there is a higher risk of emergence of zoonosis.
Yeah, Marianne, if it comes to the amount of people and amount of livestock living together on a very small area, is the Netherlands then a hotspot for zoonosis?
Well, it's one of the areas which is at risk certainly of expansion, but also maybe of true emergence. We've seen that with the Schmallenberg virus epidemic, epizootic, a big veterinary disease outbreak which had never before been seen, that started on the border of Germany and the Netherlands.
And when was that?
When was that?
It was in 2011. Oh. But I think that there was also a large role there for the invasion of new vectors that brought the virus to the Netherlands. And that has to do, that's also a very important issue in emergence of zoonotic viruses, that we have vectors that come more up north due to the global warming.
Exactly.
And these kinds of examples is what we use. I mean, from the medical side, I teach about them and say, okay, we were lucky this was not a zoonotic, that this disease did not have a zoonotic component, because there was a massive epidemic. And I've always called it the Zika of cattle. But it really illustrates that there's, across the world, there's no escape. We really have to think about this. It's not a far away, distant problem. It's something in our backyard as well.
Yeah, it's obvious that there was a slow response in public health institutions with respect to COVID, especially in those areas in the world that had not suffered from SARS. Do you feel scientists were sufficiently prepared for the current pandemic?
Well, what is prepared? I think in some elements, yes, in some elements, no. If I look at the big successes, of course, the mRNA or the vaccines, which would not have been there if there had not been massive investments in that type of research, if we didn't know which components of the virus are immunogenic, how to make these vaccines. So that's, I think, a big success. The same clinical trial networks in humans. For the first time well, I think probably in history that so many clinical trials were done, brought to conclusion because of international collaboration. So that works, and that was prepared, like you mentioned, the situation in Asia. And other elements, we are looking now at a problem with variants popping up that behave differently, that spread faster. The information comes from epidemiology, from looking at the virus genomes, and virology is saying, hey, hang on, but we need to test this, but that will take months, and that's too slow. So we have to rethink how we do that kind of virology as well.
But there are good developments also, because compared to the SARS outbreak in 2003, we were much faster. Well, the Chinese were much faster in detecting the virus and characterizing it. Yeah. And sharing it also with the world.
The COVID-19, you mean?
Yes, COVID-19, yeah. Well, there is still improvement to make, but the new tools that we use and are now already developing also for detecting the variants much faster than we did before. So there are very good developments, and we certainly have to work on that to even improve them and to think how to use them very intelligently.
And it's a similar toolbox that you use for Schmallenberg, right?
Yes.
Yeah.
Yes.
So it's, and that's
well, there's lots of improvement in making it faster.
Yeah. And viruses have also their surprises. If you compare, for instance, the SARS virus and the COVID virus, they're families. One is called one and the other called two. What you can see from the SARS virus, 2003, could only spread when people had a fever. That was easy to control, because when people have a fever, they're ill, and most of the time they're home.
And otherwise they were not susceptible.
They were not spreading. And now this virus, they started surprising us.
So the virus has changed?
The virus has changed. We have a group which remains asymptomatic, that's between the 20 to 25%. And we have people who also having no symptoms before they're getting ill, what we call pre-symptomatic. And those 2 or 3 days before, the viruses are building up and that they can spread. And these people are innocently or unknowingly, they are spreading it. And that became one of the difference. If you look at SARS, totally globally, 8,000. Now, countries have 8,000 per day.
Yeah. That's a similar parallel with smallpox. That could be eradicated because by the time people were infectious, they already were dead sick. Yeah. Same with SARS. And it's very different with this virus. But back to the science, and that you can tell if you look at where this virus binds. So looking at the receptors, the tissue distribution. So that's the kind of panels of laboratory studies that you can do very fast if a new virus pops up, saying, okay, so where does it bind? What is the tropism? What does that tell us about how this may be spreading? And that's the kind of technology that really has been developing. And I think it's really... it's exciting to bring that together.
Mm-hmm. And how do you feel scientists or science can help in prevention of a next outbreak of a new or a mutated virus?
Well, I think we really have to rethink about how we are living on this globe. Because we are living together with animals so closely, first of all, but also we have a rapidly increasing population still. And that means that there is a large demand for more and more food production, more and more animals kept.
And increasing mobility.
And that's also increasing vulnerability and also increasing contacts between animals and humans. So that means that we really have to rethink how we can change that in a way that the risk decreases.
But who or where or which scientists could pull on a string in order to influence that? Because you name like a rising world population.
It's multidisciplinary. It's a big challenge and it requires many different disciplines. That's clear. And so we are also collaborating in the Netherlands Center for One Health for that reason, saying, okay, it's no longer just about human diseases, animal diseases,
also about
environmental, wildlife diseases. It needs to be brought together.
Do you feel COVID brought like an integration of all these different disciplines? Not enough.
Not
if I look
yet.
at you.
Not yet.
No, I think that's something we need to work on.
Yeah.
Yes,
exactly. But may I want to build on this one. So what you need, national coordination and a multi-sectoral approach, but you need also an international approach. Nobody is an island. So we're all connected. Even when you're doing very well, but your neighbor's not, then you still have a problem.
COVID proved anything, it is this. Yeah.
Absolutely, and I think COVID is not anymore at that stage. But if you think about preparedness, we are as strong as our weakest link. Mm-hmm. And that's also where international cooperation comes in. That's also where it means that whatever resources you have, whatever knowledge you have, you need to share.
Yeah, but if you say so, I immediately think about vaccination strategies and the battle for vaccines. Do you feel this lesson is being integrated right now?
Well, there is some change. If I look we were discussing the test development. It took a while to get enough diagnostic testing up and running for people to have access to testing, and that was a fight for reagents, every country on their own, which makes no sense, because if you have no testing capacity in parts of the world, then you don't know what's going on. With the vaccines, at least in Europe and with the WHO, there was an attempt to coordinate who buys, who gets access, and how do you make sure that some of the purchasing power of the rich countries also is used for buying vaccines for countries that cannot afford it. So, and those are things, and also rethinking cost. We may think a vaccine $30 or euros is cheap. That's not, if you look at the global picture. So rethinking ways of producing things in a very much cheaper way, simpler way, but still, of course, effective.
That needs also something else. This is also where governments have to come in. This you can't leave to the markets, because if we think about something in the future, what is not yet there, the markets can't respond. So there you need investments. And like with the COVAX initiative that globally that's helped with not only buying vaccines, but also in an equitable way trying to share it, that is very much needed. But then governments need to be upfront ready to do so. One of the successes why we have made some progress with all the viruses is that governments this time, even though they were not yet clear if the vaccines would be working, but money was put on the table. Mm-hmm. And that was at least a very good balance to look from that risk we can also not put to the private sector. Mm-hmm. But we need also then to be able to negotiate the price.
Yeah, so what you describe is how COVID actually changed the playing field. So it called for strong institutions, for scientists having a lot of authority, the state coming in. Let's have a look finally at this university, Wageningen University and Research. What can this university specifically do in order to help prediction and prevention?
Well, this university really works in the agri-food domain. So that means, and I already explained that a lot of livestock farming has to do with food production. So when we really look at how we are doing that, there are lots of ways where we can improve the resilience of the animals. We can improve the way we are keeping them. We can improve welfare of animals. We can improve housing of animals and all that kind of stuff. And also, we can do much more on prevention of introduction of viruses in livestock. And also,
you say the way we keep our livestock, we and everybody else in the world, is already an important thing in preventing viruses to spread.
Yes. Yeah. I think we have to make those production systems more sustainable. And what we are really trying to do also in this university is to create circular economy where emissions from livestock are not simply going into the environment, but are made circular, so there is not much going out of the farming like emissions like CO2 or nitrogen, and also the risk of zoonosis can be decreased by improving hygiene in farming and by changing the context with wildlife and all that kind of stuff.
Okay, thank you. Well, that is at least a very concrete thing that for this university to focus on. We've got to leave this now at this, but we'll continue our discussion later. It is at least plenty food for thought. And in our second half hour, we will talk about the relation between virus outbreaks, human lifestyle, and health. But first, we're going to enjoy the sounds of jazz trumpetist Erik Vloeimans. And it isn't the first time Vloeimans performs on the celebration of the Dies Natalis. In 2018, he and his ensemble stood on the stage of a full Weierzaal in this building. Today, there's a large audience at distance, but that won't stop the music. The floor is to Erik Vloeimans and his ensemble. Thank you, Jeroen van Vliet, Jasper Somsen and Erik Vloeimans for this beautiful velvet sound. Meanwhile, Tessie Hick joined us on stage. Tessie developed a great interest in virology while studying biotechnology at Wageningen University and Research. After graduating, she stayed to join the Laboratory of Virology as a PhD candidate. She studies the environmental safety of self-replicating RNA vaccines. Her research can help develop a vaccine technology in which viruses are used to fight future virus outbreaks.
Have you ever thought of viruses as a useful tool? Perhaps not, as they carry so much negative connotation, especially now in the current COVID-19 pandemic. But what if I tell you that there are viruses which we can use as a vaccine tool to fight future virus outbreaks? My personal favorite group of useful viruses is the group of alpha viruses. We can use part of the genome to improve the current RNA vaccines which protect against COVID-19. These current RNA vaccines contain a fragment of RNA which is comparable to DNA. This fragment of RNA encodes for one of the coronavirus surface proteins, the so-called spikes. Upon vaccination, the RNA instructs the cell to produce these spikes, which will trigger a protective immune response in our body. We can enhance this protective immune response by adding the self-replicating genes of an alpha virus. During vaccination, the self-replicating alpha virus gene will copy its own RNA and thereby produces more spikes. This will induce a more natural and stronger immune response, even in one shot of a lower vaccine dose. Thereby, this vaccine could enable faster vaccination of the population. But is it safe to use viruses to fight viruses? Well, this technology is safe by design, as the self-replicating alpha virus RNA cannot spread from one cell to another as a wild-type alpha virus would do. These wild-type alpha viruses also encode for structural genes, which form virus particles, the vehicles for transportation from the viral RNA from one cell to another. Since the self-replicating alpha virus RNA does not encode for these structural genes, it cannot spread any further than the first cell it enters. Despite this safe by design technology, we wonder if the self-replicating alpha virus RNA can reverse into a spreading virus by recombining with a hydrologous wild-type alpha virus. So can the self-replicating alpha virus RNA mingle with a cousin from nature and form a new alpha virus? In my current research, I studied the incidence of RNA recombination between self-replicating alpha virus RNA and wild-type alpha viruses. If I detect a recombinant virus, I will evaluate how I can improve the system to make it even a safer vaccine application. Thereby, this research contributes to the development of a safe and efficient vaccine tool in which we use viruses to fight future virus outbreaks.
Thank you, Tessie. Marianne, you've been listening, you're a virologist.
Yes.
Do you feel that these self-replicating viruses are an important tool in preventing future outbreaks?
Yes, so vector technology and anything that really improves the expression of the parts of the virus that you really would like to immunize against is really, I think it's cool. I do have a question though. I mean, how are you, because I expect that we will need repeated vaccination over the years. How are you going to overcome vector immunity?
That's a very good question, yes.
Thank you.
So several papers have shown that for this type of vaccine with the alpha viruses, that's not a problem if we use the vector for the vaccine packaged in a nanoparticle. But it's a very good question to also further study if we can do this for several times, and not only for a booster vaccine, but also years later again. Yeah.
Wim van der Poel, what is the potential of this technology?
Yeah, I think you can. Well, I would assume that it should be possible to get good protection But I wondered, in your research, how can you be sure that you detect all the recombinants? Because you want to know if there are recombinants and if these recombinants are safe. So how can you be sure you detect all of them?
We cannot be sure, no. So we try to detect recombinants by this detection assay, and then we detect the recombinants which we expect. But of course, it's also interesting if we can detect different kinds of recombinants. And for that, we could use next-gen sequencing approaches, for example. But that will be a next step in this research. Yeah.
Okay, thank you.
Thank you, Tessie Hick, and good luck with this important research. With our experts at the table, I would like to talk about a healthy or not so healthy lifestyle and how that affects the pandemic. So how can science help a society to be more resilient and less vulnerable to a virus outbreak? Emily, you've been there being silent so far. You are a professor in consumption and healthy lifestyles, I should say it right. Yeah. To what degree does lifestyle now affect one's vulnerability or resilience towards viruses?
Yeah, I think it's important to realize that before the COVID-19 outbreak, the pandemic took place, we already had to deal with a health crisis, and that had to do with lifestyle-related diseases. So in the Netherlands, many people, and not only in the Netherlands, but in most Western countries, many people suffer from obesity, diabetes, cardiovascular diseases, and those suffering from those diseases increases the risk of a severe outcome of COVID-19 infection. Those diseases in itself are largely lifestyle related.
And so with another lifestyle, you would erase these chronic diseases for a large part.
Yeah, and with another lifestyle, I think we can prevent that many people develop these chronic diseases, but also prevent that once a pandemic takes place, that it has such severe consequences in society, and leading to such a detrimental health outcome.
And do you feel that policymakers or politicians are open to this message that actually public health is a crucial matter and lifestyle sciences in that respect as well?
Yeah, I think they become more open to this message, and I think that COVID-19 also brings it a bit more to the forefront that we already had a health crisis. I still think that there are major steps to be taken, both in terms of how to advance our knowledge on promoting healthy lifestyles, as well as on implementing those insights. There are some insights in how to change behavior, but that doesn't always result in the right policies taking place. And on the other hand, we still need a better understanding of how we can shift lifestyles in a way that those novel lifestyles are sustained over time and can really contribute to prevention and health promotion.
Mm-hmm. Do you agree, Henk, that COVID was a wake-up call in this respect?
Actually, it's re-emphasized the issue that we already knew, that we have a crisis on board with non-communicable diseases, but also something else. And I think sometimes when you look at the money, it gives you a picture. The Netherlands, we have a very good system. More than 10% of the GDP is being spent on health, and that's good. That's better. The Dutch are in somewhere in the middle in the EU.
But is it spent in the right way? If you look
yes, now there you come. But if you look from what has been spent on public health, that is only 0.5%. I think the balance is lost. And I do think that we need to do some rethinking. And that's also a little bit linked to something else that you have seen in the Dutch system, but also perhaps in Europe in general. If you let play the market a bigger role in the health care, then there are two areas where it doesn't fit so well, the market. And one is about safety, the things that we were talking about, that you have a good surveillance system in place and that you start investing in things what might be coming. All the discussion that we just had about the new pandemic.
For the market logic, that is only a cost.
No, they don't even see it. It's not yet there. And
prevention is very difficult to measure.
Exactly. And prevention, for instance, and that's the other point, where the market can't see, it is not good. It's not working. So this is something where the government needs to take a leading role. So those percentages needs to come by a very clear push by the government to start investing in prevention. And that's not only important for COVID, that's important for all of us now.
But for years ago, we've reorganized our health system where the market logic is leading.
Yeah.
So you're saying the system that we have is not able or is not good enough for virus prevention.
Now let me also, I'm not a basher of the market because many things have done also quite well. I think we have a very efficient system. But an efficient system might not be good enough when, for instance, when you have COVID, then you need a little bit of a buffer. We've become so lean that perhaps we should have a little bit more beds.
We've cut out all redundancy.
All redundancy are there. And I'm not saying that you can ever prepare yourself properly for a pandemic, because that's always one, but you need to have a little bit more capacity to do things. And it's including also start investing in public health.
Exactly. Because public health is not valued by the market, Emily.
No, I agree that we need to invest much more in public health, and also that we need to shift our thinking on public health.
In what
because
way?
if we think about, for instance, non-communicable diseases like obesity, the focus is very strongly on focusing on high-risk groups or supporting weight loss, say combined lifestyle interventions that take place in care, and then there are still healthcare interventions. While if you would want to shift, say, the average level of population health in healthier directions, so really health promotion, then you need to focus on a much wider group in society. You need to focus on, for instance, improving socioeconomic conditions, making sure that you reduce the health disparities that are currently really large in the Netherlands, that we create a living environment that makes it healthy, or that makes it easy to make healthy choices, and that invite to engage in physical activity, for instance. So the type of measures that are needed to promote health are different ones than currently being invested in.
On these disparities you mentioned between lower and higher income groups, I think, is income the only relevant factor?
No, it's a combination. It's a lower socioeconomic position, and that comes with a complexity of consequences. So it means a lower income, it means a lower education, but it also means that you have less means to afford housing in an area where you have a lot of green space, for instance, where you can exercise. Say the density of population density in those areas is higher,
much higher,
making it also easier for a virus to spread in those in those poor families. I would say.
And is science prepared to help governments and policymakers on this issue, or is there also a lot to do in science and research itself?
Yeah, but I think it's a little bit sad. The science on that non-communicable diseases are an issue, and that lifestyles are making a big difference. It has been with us for years. Look at tobacco. It kills. And why are we so slow?
But isn't that about behavioral change science? I mean, that's been one of the key conclusions of the pandemic as well. How do you, we know what to do,
but how to shift
what
behavior?
Does it take?
And I would say there is a lot of knowledge already there, and smoking cessation is a clear case where a lot of measures have been taking place, and it has become clear from smoking cessation that you need this large variety of measures and really a long-term investment to make sure that smoking prevalence reduces. It's still not optimal, of course, but for many other lifestyle-related factors, we aren't at that stage yet, and there's still a lot of knowledge also at the behavioral science. I would say that we need to gain, that it's not out there yet.
And how can science help create a favorable environment for these behavioral changes?
I think what is important is that we, first of all, invest in behavioral science, because it's
there's many little now.
Yeah, I think so, and I think that many of the investments and the emphasis on science is on more the technology-driven, and the life science technology also a bit medicine-focused. While if I reflect back on the pandemic, you see that we can make major advancements in science, in vaccine development, in all kinds of strategies, but changing people's behaviors and making sure that people adhere to guidelines, to corona measures, that's a lot more difficult.
Let's see what the other side thinks of that.
Can I say, because I fully agree, but I also see that those worlds are siloed, and it's really difficult to bring them together because the cultures are so different. The work so I'm interested to hear how you would approach that.
Yeah, I agree that those worlds seem silos, but I think that, and that is also Wageningen University, I think, and research is a place where you can combine both worlds, where we have and social and behavioral sciences present within. Of course, it's still siloed in the social sciences, but can be connected to all the different life science groups, whether it's food sciences, plant sciences, or animal sciences.
But you say can be, but is it?
I think that can be improved, definitely. But it's.
And if we look at the total amount of resources, let's say that amount of resources available for science will not change. Wim, do you feel that there should be more money being distributed to the behavioral sciences and then at the expense of the hard science virus research?
I think the most important thing is that we really involve social sciences in the hard sciences also. So work together to not only develop the technology, but also develop the behavior that is needed to use the technology in the right way. Yeah. And also to think about what affects our health and behave better to live healthier.
Okay.
But there is also a role for the politics, for the authorities to create an environment where people are more looking at their health and where there is more incentive to change your health, to change your behavior, to live healthier.
So would you be in favor of like a sugar tax and these kind of?
Actions like that could help. I don't really know about sugar tax, but I think there is a role for the government.
These kind of incentives
for the politics.
May I build on these things that we're just discussing? What's also very important from science is how can science get into policy? That translation is important.
So where does that block?
Now, so then you need to connect. And you need to make sure that you also come up with something what can be done. But also, I think politicians need also to be held responsible for things. The way we dealt with the tobacco industry, I think it's a shame. How can we so long still engage with an industry which actually is producing a product what kills?
But it's so harmful.
But it's so harmful. So we need also to look at that component. But what's also very important, like you said, sometimes the market can also help to get to a healthier path. With tobacco, we knew, make it more expensive, and it's for younger people more difficult to engage. Now look at the sugar tax. Perhaps we can also do a very positive tax. Why don't we make healthier food cheaper? And I think these things, sometimes the markets can also help with getting ourselves healthier.
Oh, I'm so sorry. We've got to leave this discussion right now because time is running out. And I wanted to know from the four of you, actually, discussing these things, virus prediction, prevention, what do scientists need in order to be better prepared for the future? Marion?
Systematic funding.
Systematic funding.
For the collaborative science.
Okay, that's the...
So we all have individual funding schemes. We really need
integration.
Bring it together, put a dot at the horizon, get it on.
Good, clear. Wim?
Yes, involving social sciences but also environmental sciences to work together. Well, I think all health-related scientific disciplines should work together, and I think more resources would certainly help for that.
Okay, Emily, what would you need?
A long-term vision, because I think these problems can be dealt in the typical 4-year trajectories that we get funding for. It really requires a long-term investment.
Okay, clear. And Henk?
I think investing in health is not a cost. Investing in health is investing in our future, is investing in the health of the people. So we need to get the resources also to make it happen. Yeah. And also I think here again, look at the market part again, we need to make sure that we get the benefits from the market, but that we also clear from where the market at a certain moment cannot do their work, that the government has to come in with a sufficient funding to make the good things happen.
On a
Delta plan.
On a long-term perspective, enough funding. Thank you very much. Emily de Vet, Henk Beekendam, Marion Koopmans and Wim van der Poel, because we're at the end of this table discussion. Thank you very much. Of course, thank you Danielle Gallardo-Albaran and Tessie Hicks. Thanks for your excellent pitches. And we're near the end of our program, and I'm going to give back the word now to Rector Magnificus Arthur Mol.
Thank you very much, Marcia. Those of you who've been earlier at one of our Dies Natalis know that we always hand out a major research award to a young scientist which has published the best research paper. So let's move to the chair of the jury, Professor Alfons Oude-Lansing, to inform us which major scientist has this year received this award.
We had quite a few very good submissions, and we had a lot of discussion about the quality of the different submissions, but we also all agreed that this submission stood out in terms of its quality, in terms of the contribution to the consortium, in terms of its clarity also. So this was actually for us also a unanimous decision.
Well, we discovered a new way of fixing CO2, capturing CO2 from the air through microbes. No? So these microbes can take this CO2 and produce organic compounds.
What was very special about this paper is this is a very good example of hypothesis-driven research, and I think it was also very special that this paper actually was very clearly written, also in such a way that it was understandable for the multidisciplinary team of researchers that did the evaluation.
Well, it means a lot because, of course, it's very nice to have the recognition for hard work in general, but also in this case it was a very special paper because it meant more than 3 years of laboratory work. Also, integration of many disciplines and many collaborators that were just not co-authors but also friends. On a more personal note, I couldn't really celebrate the publication of this paper due to personal circumstances, and this award has also now allowed me to revive that moment and being able to finally celebrate it.
A great winner we have for this year. Well, ladies and gentlemen, we come almost to the close of this Dies Natalis, the 103rd. Having heard all the challenges we are facing with the current pandemic, but especially with future pandemic, you might wonder, what is Wageningen University Research going to do about that? What type of research are they planning? Let's move for that to Ludo Hellebrekers, who is the director of Wageningen Bioveterinary Research. Ludo, inform us about this.
Here at Wageningen Bioveterinary Research in Lelystad, we do research on infectious diseases. Primarily infectious diseases in animals, but when these pathogens jump to humans, we investigate the zoonosis as well. With the ERASE program, Wageningen University and Research will contribute to the scientific foundation of what policymakers, decision makers and society need to try and prevent the next pandemic from occurring, or at least help and prevent the catastrophic effects as occurred with the present pandemic. ERASE stands for Early Recognition and Rapid Action in Zoonotic Emergencies. Together with many collaborators, both nationally and internationally, we will work at developing new knowledge and methods in order to prevent, recognize and combat new zoonotic emergencies in an early stage. Think of accurate pinpointing of hotspots, areas in the world where the next zoonotic emergence may occur. And think of developing smarter and more effective early warning systems and designing vaccine platforms that can help design, develop and adapt new vaccines for upcoming infectious diseases. We will take an interdisciplinary and collaborative approach from a One Health perspective. That means that we will consider the interplay between the health and resilience of man, animals and ecosystems. Together with our partners, we can make optimal use of the insights we're gathering today to be ready to answer the challenges of tomorrow.
What surprised me most as a Rector Magnificus and us as an executive board of this organization is that during this disturbing pandemic, we continued at our organizations to do excellent research and excellent education. Not in a business as usual way, but adjusted and fine-tuned to the new context and the new situation. So in closing this Dies Natalis, I want to thank and express my gratitude also to all our staff, to our teachers, to our researchers, to our supporting staff, but especially also to our students for their commitment and major effort in coping during the pandemic with all their work, education as well as research. And with that, I hope to see you next year with our 104th Dies Natalis, and then hopefully, of course, in a normal situation where we can also live with the real excitement of a Dies Natalis, having social interaction before, during and after the Dies. Thank you for staying with us. See you next year.